Welcome to part one of the audio PowerPoint for the immune system. This first section is about protection, inflammation, and the immune response, and also will cover the hypersensitivity disorders and the various infection control precautions. If you click on the immunity link below on this slide, that is an active link, and there is an approximately 11-minute video that does a nice job of explaining the immune system functions, including the external barriers, the non-specific internal response, and then the rest of our immune response. We have several functions that the immune system is responsible for, but in general, I think most people think about its response as far as infection and just that defense mechanism. We have both specific memory and non-specific, where there is no memory involved with the immune response. And we will be talking about the innate and natural immunity, which is non-specific, we are born with it, and the adaptive or acquired immunity, which is our specific type of immunity. Of course, the immune system is responsible for preserving the internal environment, maintaining homeostasis. We have to find a balance where we are protective of ourselves, but that we don't have too much immune response or an overactive immune response where we kill our own healthy cells. And this is part of the problem of what happens with the autoimmune disorders that we will be talking about at a later time. Surveillance, of course, we have cells, very specialized cells and cells that do contain memory that are able to recognize and maintain the body from unhealthy factors, or that's what it will try to do, prevent development of malignant cells or cancer. This is an excellent slide just summarizing the 3 lines of defense as far as our immune system goes. The first line of defense is the mucous membranes, the skin, which is our largest organ of the body. Again, we have very specialized epithelial cells in the skin. We have cilia, we have nose hairs, we have ear hairs, all of those sorts of things that also help with protection, and we will be talking about all of those more specifically in here in a minute. The second line of defense is primarily our inflammatory process, and we know that with every response to trauma or illness or infection, the body will respond via the normal inflammatory response. We'll talk about that is primarily where we have neutrophils and macrophages, which are mature monocytes that basically will go to the site of injury or trauma and act fast as far as protecting the body. So that phagocytic action of white blood cells is very, very important here in the second line of defense as far as the inflammatory response. Again, primarily neutrophils and macrophages. And then we'll talk more about what a fever can do for us as well. And then the third line of defense is a specific. It's a adaptive or acquired. We have 2 types. We have the humoral, which is the antibody-mediated response, which involves our B cells, and then we have a cell-mediated immune response, which is our T cells. Again, both of these are specific, meaning that they do have memory. So, for example, if I have an antigen that invades, my body forms an antibody to it. The next time, if I were to see that very same antigen, my body would automatically have memory and respond. Another important piece to understand is that we have what's called human leukocyte antigens. Basically, we have surface proteins that are unique to our own self and our own individual. So this is going to be how our body is able to identify that the microorganism is a foreign invader. So I just like to think of it as a team uniform, and I have a team uniform with my number on it, and somebody else has a team uniform with their number on it. And so my body knows all of my cells with my team uniform and my number. And so that's where it will recognize antigens or foreign materials that do not have my same uniform on. And again, it's almost like, too, if you think of a very specific barcode, it knows that barcode and is going to be very specific, excuse me, to that. On the last part of the slide, it talks about stimulating the immune response. And you see the antigen there, and that's an antibody generator. That's how it gets its name, antigen. So as an antigen invades, it will generate an antibody to protect the body against that antigen now, and then again it will have memory to do so in the future. This slide will review the basic A and P for the immune system. There are three major components of the immune system. One very important component is the bone marrow. The bone marrow is responsible for production of white blood cells and storage of the mature B cells, also storage of the stem cells. All of that is very, very important for our immune response. The bone marrow, as you remember from hematology in the cardiac unit, is also responsible for production of red blood cells and also production of platelets. So we'll talk about how important the bone marrow is to us for hematological type conditions and for maintaining our immune response. If we have bone marrow suppression, we would expect that we would have decreased red blood cells, decreased white blood cells, and decreased platelets. And of course, we know what kind of issues we can have with that. The white blood cells, of course, are a huge part of our immune response, and they are the cells that are going to help to provide us protection. We will be talking about all the various different types of cells. So far, we've talked just briefly about the immune response, the inflammatory response, which is the neutrophils and the macrophages. Those are two types, but we will also be getting into the T cells and the B cells and their various functions. The thymus is responsible for production of the T cells, and the bone marrow is what's responsible for production of the B cells. So again, we will be talking about that as well. So besides the bone marrow and the white blood cells, the third major component of the immune system is our lymphoid tissue and the lymphatic system. So we know that we have lymph nodes that are distributed throughout the body. They connect the lymph channels, and their job is to remove foreign material before entering the bloodstream. So our lymphatic system does a really good job of removing that foreign material before it ever gets picked up in the blood and causes worse problems and or infection, cancer, etc. We'll talk about metastatic cancer and that's why they check the lymph nodes closest to the site of the primary source of cancer, and if that cancer has gained access to the lymphatic system, then it has likely been spreading to other areas. So again, we'll make some connections when we get into that area as well. Next on the slide talks about the spleen, and the spleen is very, very important it is responsible for old red blood cell destruction, but then also it is responsible for helping those B lymphocytes to develop into mature cells and to store them there, storage of mature B cells. So the spleen, while we can live without a spleen if we have to, it is very important because it's a dormitory for those mature B cells, and then also helps with the destruction of the red blood cells and getting rid of those materials. So the spleen is again very vascular, and we'll talk about if you have a spleen laceration, very similar to when we talked about if you had a kidney laceration. Those would be things where the patient can really have problems with bleeding internally. They'll probably be on more of a strict bed rest as to not activate any bleeding, and you'll more than likely see hemoglobins drawn like every 8 or every 12 hours, and these are mainly like traumatic car accident type conditions, or it could be gunshots and wounds that way too. So we'll go into more depth about all of our various lines of defenses. This first line of defense is pretty straightforward. This is our skin and mucous membranes. This is all of the external environment things and all the external things that we have in our body, and even internal structures and features that help us with that very first line of defense. So for example, you'll see lysozymes in the tears. So that's a chemical enzyme that is in nasal secretions and tears. So if you've ever had something in your eye, you feel like you have something stuck, you have sand, gravel, whatever, our eyes start to water right away. And that again is that chemical that is trying to rid that foreign material out. Of course, we know our skin is our very best source of defense for a physical barrier, provides a physical barrier. We also know that we have sweat glands and that perspiration is acidic, so that helps ward off anything on the skin as well. Mucous membranes, we have mucous membranes everywhere, and that defends the body's openings from invasion. For example, reproductively wise, which isn't on this picture, we do the vagina, which is a large mucous membrane, does protect against invading organisms, and we have very acidic vaginal secretions, especially during childbearing years, to help with that protection of that opening. If we go to the right side of this picture, we think of the cilia, and that traps any of the inhaled debris. Those are our nose hairs, and it traps it until we basically either sneeze it or cough it out or something. And then the GI tract, we think of our stomach and the actual acidity of stomach contents, and that's going to help with not only digestion, but it definitely helps with breaking down any ingested bacteria. For example, if we know that there are microbes on like fresh fruits and vegetables, depending on how well things were washed, etc., we may ingest some of those microbes and bacteria. Our stomach usually is going to take care of that with the acidity and the hydrochloric acid and gastric acid to make sure that we don't get sick from that. But certainly, food poisoning can happen, C. diff, etc. There's other things that can happen in our GI tract, but our body does a pretty good job of helping to protect. And then the urinary tract, we know that the acidity in the urine is going to help with flushing out any bacteria that are trying to gain access. Try to keep increasing fluids, etc., and keep that acidity where it needs to be with, say, like cranberry juice, in order to make sure that we're flushing things out that shouldn't be there. The second line of defense then is our inflammatory response, and we'll talk about the white blood cells that work to help us with that. They work in conjunction with the plasma proteins. The plasma proteins basically activate the complement cascade, and that is what helps to signal those phagocytic actions to be more effective and efficient. We're not going to go a lot into the complement cascade, but I just wanted to explain what that's about. The phagocytic white blood cells is really what's most important. We have neutrophils, and then we have that are very fast acting white blood cells. They're the first to gain access to a site of trauma. Within 90 minutes, they're going to be where they need to be. Monocytes are going to mature into macrophages. So again, that's also an independently phagocytic cell that's able to ingest, engulf, and destroy and get rid of bacteria, viruses, fungus, other things that we don't want to be there that are invading. So neutrophils and the monocytes, or what we know as mature monocytes, our macrophages, are going to be our two key white blood cells that help us with the inflammatory response, and we're going to be talking in more detail. And then, of course, we'll also have some slides on what a fever can do in the inflammatory process. So far, we've talked about the first and second lines of defense, and both of those are innate or natural immunity, meaning that we are born with them. So we have a genetic predisposition to what our immunity is. This is present before any exposure to pathogens occur. It is nonspecific, so it does not have memory. It is immediate and can be very effective, but is more short-term, and it is not an adaptive response, meaning it is not acquired. It is just genetically innate in us. Does not provide us true immunity with the repeated exposure because there is no memory. So again, this innate natural immunity includes our skin and mucous membranes and those things that we've talked about on the prior slide, and then the inflammatory response as the second line of defense that is also natural or innate, and again, that's just the activation of the phagocytic response. The third line of defense then is the adaptive or acquired immunity, and this is immunity that we develop over time during our lifetime. This is very specific and does provide memory and long-term immunity. So it's not genetically determined. It is based on when we have exposure to antigens, microbes, toxins, or any foreign substance that we consider to be an antigen, our body will form a response to that, and then it will keep its memory to that pathogen for subsequent exposure as well. So white blood cells are central to both a natural or innate immunity and also to acquired or adaptive immunity. I just like how this visually shows you that we have our lymphoid stem cells, which is basically our lymphocytes, which are our B and T cells. And then we have our granulocytes, which are our neutrophils, eosinophils, monocytes, which you can see here turn to mature macrophages. So those granulocytes and the lymphocytes are what we're going to be looking at when we look at lab work on our patients, and they have a white blood cell with differential. The differential is going to differentiate all the different cells, the percentage of those cells as they compare to each other, and what percentage they're normally supposed to be. This is also just mainly to show you that all of these white blood cells have different structure, shape, color, etc. So under a microscope, they can really dissect and see numbers and quantities of cells in proportion to other cells. And again, for our standpoint, we're not necessarily doing that, but this slide also just sort of shows you what the main job or the primary function of the different types of cells are, which we will be getting more to. Next, we will talk about the sequencing of the inflammatory response. Again, this is our second line of defense. It is a non-specific form of immunity. It's the second line of defense after our skin and mucous membranes. So I know we've talked a lot about when patients what would cause an inflammatory response to be activated. And I just want to start there. Tissue and cellular damage can have a variety of causes, including infection, trauma, surgery, oxygen deprivation or ischemia, nutrient deprivation, could be genetic or immune defects, could be chemical agents or temperature extremes such as burns. So those would be just some examples that could cause the inflammatory response to be activated. And I think it's important for us to recognize when we would expect to see an inflammatory response. Certainly we've talked about it when patients have surgery. You'll have an inflammatory response when you get stung by a bee, when you have a sore throat. Pretty much any condition, if we think of arthritis, we'll talk more about that when we get there as well. We see things primarily in 3 stages as far as the inflammatory response. Stage 1, anatomically and physiologically, is the vascular response. This is when the tissue injury or invasion basically activates our mast cells, our macrophages, to start the whole complement system and complement cascade. There is release of histamine and other chemicals, kinine, serotonin, which cause dilation. So that dilation increases the blood flow to the area, to the localized area. So increased blood flow to an area is going to cause that to feel warm. It's also going to cause it to look more reddened. So when we think of red and warm or red and hot, that is a result of the hyperemia or the increased blood flow, increased concentration of red blood cells to that area, causing that red hue and that warm or hot temperature. And then the edema is going to come because we end up with a capillary leak. Increased hydrostatic pressure in the capillaries will produce that fluid to leak into the third space. So again, there will likely be localized and or systemic edema based on what the inflammatory response is doing, what stage it is at. So again, I'll challenge you to always be thinking about the physiology behind our assessments, and hopefully now you have a better understanding of why we expect red, warm, and swelling or edema with inflammatory conditions, and which then could develop into signs of infection after, that 7 to 21 day period is typically when infection will set in. Stage 2 is neutrophilia, which is mature neutrophils will increase. This is where those mature neutrophils will go to the site of infection, inflammation, trauma, and the neutrophils you can think of like soldiers with guns. They are at the front line. They are the first, they are the fastest, they are the quickest to get there, and they're going to respond very effectively together as a team to fight off that infection or that foreign agent. And then we'll talk more about the phagocytosis, and again, this is primarily just thinking of engulfing and eating up any of the foreign material. So neutrophils and macrophages are phagocytic, and they will go ahead and engulf and destroy the foreign materials. And then stage 3 is the tissue repair stage. Again, this is where we are going to have fibrin that's forming that will trap some of the microbes for removal by phagocytosis, but also formation of clot and helping with stopping the bleeding, helping with that portion of the healing. And then of course during that tissue repair, scar tissue is going to form to help complete that healing process. This is just a different version of going through the inflammatory process and the steps of phagocytosis. So initially there's going to be exposure and invasion, and then the damaged tissues secrete chemotoxins, toxins that attract the neutrophils and the macrophages that release the debris and then bind to the surface antigen. Adherence then will happen, and this is where the binding of the phagocytic cells to the surface antigen happens. And then there's the recognition of the HLA. Again, that's the human leukocyte antigen. That's your uniform. That's the identification of non-self. And then cellular ingestion or engulfment, phagosome formation, which is where the granules will break and then release that enzyme that as far as the particles and the debris of what was engulfed. And then degradation is again engulfing that target. And this is even a more simple version, which I really like, and it just says when foreign material or foreign matter such as bacteria, dead tissue, comes in contact with the cell membrane of the neutrophil, the membrane surrounds it, and you can see basically pinches off the area, leaving the membrane intact, and then that engulfed material is left in the vacuole of the neutrophil, and the enzymes will destroy that foreign material. The next two slides then will talk about the cells that are primarily responsible for that second line of defense, the inflammatory response. So the neutrophils, the polymorphonuclear is the PMN, and that doesn't matter, but they have many shaped nuclei, which is why they are called polymorphonuclear. But the neutrophils are the very first white blood cells, the very first type of white blood cells to go to the site of microbial infection or injury. They usually are there within 90 minutes of injury. They have a short lifespan of like 12 to 18 hours. And if they're working during a very active infection, they might only live for a couple hours at a time. But the maximum lifespan is really that 12 to 18 hours. So my best analogy is thinking of the neutrophils as our soldiers that are in the front lines with their weapons. They're going to go there, they're going to work, they're going to be all of them together, going to provide a really great great front line of defense for the body. The phagocytosis then will happen. The neutrophils are able to phagocytose all on their own and remove and get rid of a bunch of the debris. Death in large numbers of the neutrophils will form pus. So if you see pus, and again in wounds and things, we don't necessarily like to see pus or purulent drainage. That means that there has been a bunch of dead neutrophils that have compiled in that area. Most common cell type in acute stage of inflammation. So again, this is the main cell type that we're watching for. This is the big one that we're going to be always looking for when we start talking about this white blood cell with differential. We would expect the neutrophil percentage to go up after acute inflammation, trauma, injury, because we're expecting the neutrophils to respond and go to that site first. The monocytes, then we know monocytes will mature into macrophages. So if you think of monocytes and macrophages as the same thing, that is fine. The stem cells, like I said, form the monocytes, but then those are released and will develop into mature macrophages. The macrophages then are have the phagocytic action. Monocytes or macrophages are larger. They are slower to respond. They have a longer lifespan, several days for a lifespan, and you can see whereas neutrophils got to the site within 90 minutes, the macrophages may take 8 to 12 hours. So I like to think of the macrophages as their tank in the war. They are slow moving, but they are big and they're efficient and they are unstoppable. So that's really important to think of it being longer lasting. They will also basically send messages. The macrophages will have phagocytic action all on their own, but they will also present the antigen to the T and B lymphocytes to basically activate that adaptive or specific acquired immunity. I like how this picture shows you that the phagocytes eat the bacteria, but parts of the bacteria, the antigen, goes to the surface of that phagocyte, and the phagocyte then actually presents that antigen to the T cell, because we want the memory to be happening, so that's how they will activate and kind of bring in that third line of defense where we'll have some specific immunity and memory against that surface antigen. What I want you to focus on this slide is more off to the right side. We don't have to really go into all the detail of the activation of the complement cascade, but I do like how it shows at the top the vasodilation is going to happen initially with inflammatory response. We will have increased blood flow to that area where we'll have erythema, redness, and the warmth. And then the next one talks about the vascular permeability, where that capillary membrane leaks fluid, and we will have leakage of fluid into that third space, so we'll have that localized edema. And then the cellular infiltration, this is where we have increase in the neutrophils that mobilize additional leukocytes, but again, that phagocytosis of fluid and debris will form pus. And then the thrombosis and stimulation of nerve endings or pain comes next. We talk about the tissue repair, the fibrin that forms the mesh to trap the microorganisms for removal, and then that fibrin that also helps to stop the bleeding. I like this slide because it's basically talking about the 5 cardinal pillars or manifestations, signs and symptoms of the inflammatory response. So you can see heat, redness, and swelling, which we've already talked about. That heat and redness as a direct result of the vasodilation, that vascular response. That vasodilation happens as a result of histamine and other kinines and chemicals that are released and increases blood flow to that area, causing the heat and causing the redness. Swelling is a direct result of the increased capillary permeability. As the hydrostatic pressure increases, that pushes fluid out, and so you'll have an outpouring of serous fluid, proteins, albumin, etc., into the interstitial space. And then pain, caused by pressure of the accumulating fluid on the nerves, and also those chemical mediators could irritate nerve endings, so pain could be associated as a cardinal feature of the inflammatory response. And then oftentimes you'll see loss of function, decreased motion of the area that is also affected. Now again, remember the inflammatory response, it starts and activates from the onset of the injury to approximately 72 hours. That's when we're most going to see these symptoms to occur, in the first 72 hours. And again, you'll hear over and over that very first 24 to 48 hours, that's going to be our maximum time for swelling, redness, pain, etc. This is going to be where they really want the patient, for example, if it was an ankle injury or fracture or something, to be in bed with that leg elevated, iced, etc. Local versus systemic response. When we think of the inflammatory response, we think a lot of the localized signs and symptoms, the localized redness, heat, swelling, etc. at the site of injury, the pain, the loss of function or motion. And again, this remains a cellular injury site when it stays localized. But we also know that the microbes can survive that initial phagocytic inflammatory response and end up producing a systemic response, a systemic infection that can reach the blood, that can reach the lymph nodes and spread throughout the body. So fever, while fever doesn't prove that it's a systemic response or an infection, this is definitely going to be a very common sign of a systemic response, that we are expecting the possibility of systemic infection or a blood-borne sepsis. Patients, in addition to their fever, might have chills, malaise, real tired, anorexia, not eating well. These are all things to be watching for. Leukocytosis then is increased white blood cells, and neutrophilia is the increased neutrophils. So if somebody has leukocytosis, of course they're going to have that. It's an inflammatory response. So there's going to be more white blood cells that are going to be messaged to be sent. But the left shift has to do with the number of mature versus immature neutrophils. So left shift will be when there are many fewer mature neutrophils or white blood cells, and so we have an overabundance of immature white blood cells trying to fight and be a line of defense. And then the plasma protein synthesis, again remembering that we have increased fibrin, blood clotting, etc., that is going to be helping during that localized inflammatory response. A fever is our body's best natural defense against an invader that may be susceptible to warmth. So some invaders will die at higher temperatures. So this is the idea behind our body, our core body temperature wanting to get hot, and why we would run a fever, because hopefully that internal defense can fight off some of those invaders that will not survive because of the high temperature. So that's the nuts and bolts behind the actual action or physiology. We know that fever is not a specific sign or response of a systemic infection. It's just another clue that may indicate that a systemic infection may be getting worse or maybe gaining access to the bloodstream. Again, be aware that a fever in healthy people can be a good defense mechanism, but we don't want to mess around with fevers in infants and young children, for example. We would treat them promptly. But for some, if you are not in that age group and you are running a fever, it is a good way to let our body run its own course and let the body actually fight off the invader. Instead of taking fever reduction medications, it sometimes can be very ideal to let the body rid itself and just to go through the stages of the fever. But again, I want to reiterate, infants and children, we don't want to mess around with a fever. There's the possibility of febrile seizures, which of course we would not want to happen. So a fever helps by stimulating white blood cells to destroy invaders or limit the growth. It, of course, decreases the available free plasma iron. And what this does is it decreases the iron in the blood, which then slows the reproduction of the pathways. And then it also is responsible for introduction of interleukin-1, and that's a protein a pro-inflammatory cytokine that increases the amount of interferon. Interferon is what travels to other cells to increase resistance to the invading virus or organism. So one thing I want to mention before moving on is that the fever alone, again, does not 100% mean it's a systemic infection, but it's just another sign that would validate the likelihood of that. Also, elderly are less likely to run fevers, so we're not going to see that typical presentation. And then the other thing about the fevers that I forgot to mention is that you want to be cautious of febrile seizures. So that's why we would want to respond to fevers in a timely manner, especially for the high-risk age groups. And also wanted to reiterate that we understand that febrile seizures are going to be more oftentimes produced by how quickly the temperature rises, not necessarily how high the temperature is. Now on this slide, it's just another time to remind us how elderly can have atypical signs of infection, and during the inflammatory response, elderly may be more confused, maybe more sedate, change in level of consciousness, and in addition, loss of appetite, etc. They definitely may have a decline in function and be at higher risk for incontinence and falls. But again, we just have to be watching for and be very straightforward to what the signs and symptoms may look like so that we can try to identify them sooner than later. I think we've pretty well already identified the differences between the infection versus the normal inflammatory response. We'll keep talking about it. We know that everybody can have an inflammatory response. Typically, if it's, for example, a surgical wound, we're not going to expect any onset of what may be an infection until like 7 to 21 days. So in the acute hospital setting, a lot of people are more likely to have inflammatory response versus infection. Obviously, it depends on what they're there for, etc. So we're now going to get into the white blood cell count and differential. This first slide just talks about the normal WBC, 4,500 to 11,000. Obviously, those ranges can vary slightly from source to source. If we're getting above 20,000, we would most likely call that leukocytosis or that elevated white blood count. Elevated, again, does not mean that they have a systemic infection, but it is certainly more likely to see in those that would have a post-op infection or other type of infection from another cause. Again, some bacteria are strong and may resist the leukocytes, so infection can resist treatment and spread, for example. So we're going to talk a lot about what we're actually looking for as far as the white blood cell differential, how much percentage of cells, the different types of cells that we are expecting to see. I think initially it's very important to remember that leukocytosis is a white count that is elevated, and leukopenia would be it's decreased, and that means that they would be more apt to be at risk for infection, develop infection. They would have what's called neutropenia or the decreased neutrophils, and may need to be on neutropenic precautions or protective isolation, for example. In your study guide, I have created a table specifying the white blood cell with differential that is more detailed and probably more helpful to follow along than the next couple slides. But so I'll refer you to that. We will be looking at all of the different cell types and the importance of each. In particular, with the white blood cell with differential, know that the differential part is all of the other white blood cells in total. They must equal 100%. So when you're looking at the differential, you will see all of the cells listed, such as neutrophils, eosinophils, basophils, lymphocytes, etc. And you'll see a P version of that, which is the percentage of those, and then you'll see those same items listed again with an N behind them, which is just the number. For our purposes, it's really just easier just to look at the percentage, and the numbers are going to be relative to the percentage anyway. So if the percentage was high, for example, the total number would be high also typically. So again, that's one way to sort of simplify it as a nurse specialist or generalist. And unless you're working very specifically on hematology oncology, there might be reasons to look a little bit more separate. As far as the neutrophils go, they also can be broken down into mature or segmented neutrophils. And those segmented are the neutrophils that are fully mature and ready to fight. And so I quantify those or qualify those as like our soldiers at the front lines with their weapons. Immature or band type neutrophils are basically soldiers without weapons on the front line. So those aren't going to be effective cells for our body to fight an infection or help with an inflammatory response. So we certainly don't want to have more immature than mature neutrophils. So that's something that we'll be looking at here coming up. We will also later on look at absolute neutrophil count, which is utilized highly in oncology. Well, they will look basically at the percentage of neutrophils multiplied by the percentage of bands or immature, and they'll get an equation representing the total number of fighting cells. So absolute neutrophil count is very helpful and specific to helping to determine the real risk of infection and how serious it is as far as the number of available mature neutrophils comparative to the total number of neutrophils.